Right, we are starting a new unit on DNA. It comes from chapter 8 in your book called From DNA to Proteins. And basically, we're going to cover a little bit about the history of DNA, its discovery, about its structure, and how the central dogma works. In other words, how does DNA code for your proteins? And so 3 big processes that we're going to cover is DNA replication, transcription, and translation. It can get to be a little technical sometimes, so really pay attention and go through your notes carefully and do all the exercises that you're asked to. But for this first part, the first learning target is to identify the structure of DNA and explain how it can be copied as well as serve as a code for making proteins. And of course, your proteins are what give you your traits. But we're going to start first by backtracking a little bit to just explain the key contributions that different scientists made to the discovery of DNA before we get into the structure in the next set of notes. So the first individual who made a contribution is Frederick Griffith. His main discovery is that he discovered what was called the transforming principle. And he experimented with bacteria that cause pneumonia. They're called pneumococcus bacteria. And there's 2 forms. There's a form that's deadly, called the S form, and that's smooth. And then there's a R form for rough, and that's not deadly. It will not cause pneumonia, whereas the S form does cause pneumonia. And so he did an experiment that involved these bacteria and mice. And so he basically injected the live S bacteria, remember that's the deadly form, into the bacteria, or into the mice. And look at, unfortunately, you see a little dead mouse there. They did not fare well. But then he injected the R form, the rough form that is not deadly, into the mice, and they were just fine. So then what he did is he heat killed the smooth bacteria, okay, the deadly kind, and he injected it into the mice, and lo and behold, they lived, right, because he killed off the bacteria. What he did, though, next is he mixed the heat-killed bacteria with the live rough bacteria. Remember, heat-killed do not kill the mice, and the R bacteria do not kill the mice. So he mixed that combination together, then injected it into the mouse, and lo and behold, they died. And when he took samples from them, he found that there were the S form, the smooth form of bacteria, inside the mice that were dead. Well, how can that be? What happened is that the rough form was transformed into the S form. So now they were deadly. So somehow they picked up something that caused them to now have this new trait of being deadly. Well, he did not know anything about DNA at the time, Frederick Griffith, so he just called it the transforming principle, the transforming material. And today, basically he discovered, back with what he did, what we call the process of transformation, how you can take DNA from one organism and kind of insert that into another organism and cause them to have a new trait. And so what you're seeing here is a little glow-in-the-dark mouse. They took the P-glow gene that's from a bioluminescent jellyfish, and they injected it into a mouse embryo. And as that embryo started to divide and grow into a mouse, newborn mouse, it had the trait, because it now had the DNA, it had the trait of glowing just like that glow-in-the-dark or bioluminescent jellyfish did. So the next group that kind of took off from where Frederick Griffith left off was Avery, McLeod, and McCarty, a team of 3, and they discovered that DNA is the transforming principle. Okay? How they did that is they took the lysed bacteria. Remember, lyse means to break apart? We've had that term before. So they kind of busted apart the smooth bacteria, and they subjected it with a protease. That's an enzyme that destroys protein. Remember, anything that ends in ASE is an enzyme. And they also subjected it to DNase, an enzyme that destroys DNA. Well, only when they used the DNase, that enzyme that totally destroyed the DNA, that prevented the transformation from happening. So they are basically showing then that DNA is the transforming principle that Frederick Griffith discovered. And it's the DNA that our bacteria picked up and converted it into like a smooth bacteria that was now deadly. And so here's kind of their picture where they broke open and the smooth bacteria there, and they subjected it to protease and to DNase and added it to the rough bacteria. And when they just only killed off the protein, the mouse still died because it still had the DNA to pick up and be transformed into the bad bacteria. But when they killed off the DNA, destroyed it, now the mouse could live because now that rough variety did not pick up the DNA from the smooth bacteria. It was not transformed. And that's the pictures of the three gentlemen that worked together on that. Alfred Hershey and Martha Chase further confirmed that DNA was the genetic material. They used a different approach, though. They used what are called bacteriophages, or bacteriophages. You may have heard it pronounced both ways. These are viruses that infect bacteria, okay? And a virus has a fairly simplistic, sometimes, arrangement. And these bacteriophages, they basically have a protein coat, and that contains a component called sulfur, sorry about that. And then inside it, it contains DNA, which of course has phosphorus in it. So what they did is they radioactively labeled the sulfur protein coat, and they also radioactively labeled the phosphorus in the DNA to try to track which part actually enters the bacterium. When a virus infects the bacterium, what part are they injecting into the bacteria? Their protein coat or their DNA? And they found out that it was the DNA that entered, not the protein. So here's their experiment. They took they radioactively labeled the sulfur coat and then radioactively labeled the phosphorus in the DNA. They blended it up in a blender experiment, and then they centrifuged it, spun it around to see which parts, once the viruses had injected their material into the DNA, then they spun them around to separate the bacteria from the virus, and they found out that the ones that the part that actually enters the bacteria, if you see this picture off to the right here, the DNA is what entered the bacteria. The protein coat doesn't, because you notice this one doesn't have any new DNA in it. This one does have new DNA in it because the virus injected their own DNA in there. Okay? So they confirm that DNA is the genetic material that gives organisms that new trait. Okay? From there we have Erwin Chargaff. He looked at a variety of plant cells and animal cells and so on, and what he kept finding is the amount of adenine would always be in the same proportion as the amount of thymine. And likewise, the amount of cytosine was in proportion to the amount of guanine. Well, what that led to is what we now call the base pair rules, sometimes called Chargaff's rules, because he's the one that came up with it. That A always bonds with T and C always bonds with G. Now, this is going to be very important when we talk about the structure of DNA and we talk about DNA replication and transcription and so on. You need to remember these base pair rules. A with T, just think that you have an awesome teacher, right? No, I'm just kidding. And then you guys are both all cool guys and cool gals. So C with G and A with T. And then we have Rosalind Franklin. She used X-ray diffraction to show that DNA had a helix shape. In fact, photo 51, which we might have you watch after we're done with the unit, really showed that when it shows on an X-ray this kind of X pattern, it means there is a helix shape to it. So it was very important in kind of leading to the discovery of kind of the shape or arrangement of DNA. And she worked with Maurice Wilkins in his lab, but she's the one that really did all the work because he was kind of away on a sabbatical at the time and when she made a lot of these discoveries. And finally, we have Watson and Crick. James Watson, Francis Crick. They're the ones that presented the first model of DNA and showed that it was a double helix. That means two strands that were wound up in a helix shape. And they obviously had to use information from Chargaff as far as how the bases paired up, from Franklin in terms of how that it's a helical shape, because when they presented the first time, they were way off. And so by using some of the information from other individuals, they were able to come up with this first model of DNA. And then they won the Nobel Prize for their discovery along with Maurice Wilkins. Remember that Rosalind Franklin worked in his lab. We always, historians always wonder, would Rosalind Franklin have also shared in the Nobel Prize? She unfortunately died from cancer, probably because she was around all those X-rays all the time. And so it was Maurice Wilkins who shared with Watson and Crick in terms of their Nobel Prize winning. So let's do a quick little review here to the scientific contributions. We're going to draw lines connecting the scientists with their discoveries. So discovered the helix shape of DNA using X-ray diffraction. Remember, that was Rosalind Franklin, okay. Discovered the transforming principle using mice and bacteria. That was Frederick Griffith. Confirmed that DNA was genetic material using bacteriophages or bacteriophages. That was Hershey and Chase. They're the ones that worked with the virus and DNA and the bacteria. Discovered the base pair rules. We discovered him. That was Erwin Chargaff. Remember, we sometimes call that Chargaff's rules. Confirmed the transforming principle was DNA using enzymes in mice. Remember, they picked up where Frederick Griffith left off. That was Avery, McLeod, and McCarty. And then presented the first model of DNA showing it was a double helix. That was Watson and Crick. So that's just an introduction to the discoveries. Next, we're going to talk about the actual structure.